Ladies and gentlemen, you are all welcome to our YouTube channel. So, in today's video, we are going to look at the inborn error of carbohydrates metabolism. But you need to look at what the inborn error means. So, the inborn error are the group of rare genetic disorders in which the body cannot metabolize food components normally. So, generally, we know that in a chemical or in a body system, we have what we call metabolisms, which is the sum total of all biochemical reactions that are taking place in the body. So in a situation where we have, for example, of course, we know that metabolisms is a multi-step reactions. It's a multi-step reaction where A combine to produce B, and then B will proceed to produce C, and then C will proceed to produce D, from D to E, depending on the number of steps of a particular pathway. And of course, each and every step of the reaction is catalyzed by an enzymes. Each and every step of the reaction is catalyzed by an enzymes. Each and every steps of the reaction is catalyzed by an enzyme. So like, for example, now, if we can see clearly, we have a number of steps here where 1, 2, 3, 4. So we have like 5 different steps. So in the metabolic disorder, or in this inborn error of metabolism, the disorder is usually due to or is caused by a defect in enzymes. It's usually caused by a defect in enzymes. So when there is an enzymes defect, then there is a likelihood that the metabolic pathways that break down that very essential biochemical component will be blocked. Like, for example, now, if this is, we have, let's say this is step 3, we have enzymes 3, and of course, this is enzymes 4. So it means that at this stage, let's assume the enzymes 3 here is defective. So what will happen? So it means that C cannot be converted to produce D. So if C cannot be converted to produce D, then what happens? It means that there will be a deficiency of the product, which is D. The D will be deficient, and of course, then the C will be accumulated. So as a result of that colossal accumulation of C, then it will lead to a lot of medical conditions. It will lead to a lot of medical problems. So now, as we said, it is an enzymes defect, and this enzymes defect is usually due to mutation. It's usually due to mutation. It's usually due to mutation, as we know that actually from the concept of central dogma of molecular biology, we know that the DNA, we have a DNA. So DNA usually converted into RNA, and then the RNA will be converted into protein. So actually, if there is a mutation, in the DNA, then what will happen? So if there is a mutation in the DNA, it means that, of course, let's say the mutation is at a gene that is responsible for producing, let's say, enzymes 3 from the previous demonstration. So what will happen? Enzymes 3 is that it has a gene from the DNA here. So if the gene responsible for expressing this enzymes 3 is mutated, and there is a probability that there will be a production of the defective messenger RNA. So once you have defective messenger RNA, it means that there is a likelihood you will have a defective protein, which is the enzyme. So once you have a defective enzyme, it means that the enzymes may likelihood, although that in some cases mutation can be positive, because you can have a situation where mutation can increase the activity of the enzymes, and you can have a situation where the mutation can decrease the activity of the enzymes. So usually in inborn error of metabolisms, the mutation is actually a negative, because the enzyme's activity is usually distorted. So therefore, it cannot convert the biochemical component into the product. So that is an inborn error of metabolism. We should understand that there are series of steps. In metabolic pathways. So if there is a defective enzymes in any of the pathways, so there is a likelihood there will be a blockage of the production of the product, which actually or subsequently lead to the accumulation of the substrate that is combated by that particular defective enzymes. So that is an inborn error of metabolism. So what is the metabolic disease? So according to the Garrett's hypothesis, he actually gave this demonstration where he actually demonstrated that if you have A as a substrate, which convert it to produce B, and then B will now actually be converted to produce C. So now what happened if there is a problem? Let's say the enzymes here that convert B to C is defective. So what happened? It means that the C will not be generated. The C will not be produced. So if C is not produced, what will happen? There is a likelihood that the substrates can be accumulated to produce maybe a toxic metabolite which can cause inflammation, which can cause a lot of medical condition, necrosis, and so many other things. And of course, since the enzymes is not converting the product, then what happens? Then the substrate will be in excess. So this is actually Garrett's hypothesis, where the substrates are being actually blocked from converting into the product. So if there is that blockage, then the product of that particular reactions will be deficient. So there will be deficiency of the product, accumulation of the substrates, and then the production of the toxic metabolites. So then, so now, according to the metabolic disorder, what is the metabolic disorder? Then, of course, according to the Garrett hypothesis, there is a genetically determined metabolic disorder in which a specific enzymes defect produce a metabolic block that may have a pathological consequences at birth. Like, for example, we have a situation where if these enzymes is actually genetically defective, or if the enzymes are genetically defective, then there will be a biochemical disorder, and that biochemical disorder is caused to the enzymes defect, and as a result of that, there will be a metabolic blockage. And that metabolic blockage can lead to pathological consequences even at birth. Like, for example, in a situation where we have phenylketonuria, or in later life, like, for example, diabetes mellitus. So this metabolic disease is also called enzymopathy, or you can call it genotrophic disease. So this is what we call metabolic disorder. So then, what are the classification of metabolic disorder? Metabolic disorders are classified into 4. So the main causes or the problem associated with the metabolic disorder: number 1, we have mitochondrial inheritance, we have autosomal recessive disorders, we have X-linked inheritance, and we have autosomal dominant disorders. Like, for example, for the mitochondrial inheritance, we have a disease called Leber's hereditary optic neuropathy. So how do we actually have this mitochondrial inheritance? So, ladies and gentlemen, you should understand that this concept is very, very important. We know that in a cell, we have 3 components of the cells where DNA are found. So we have DNA in the nucleus, we have DNA in the mitochondria, and we also have DNA in chloroplast. So these are actually 3 major parts of the cells where DNA are found. And of course, we know that in the DNA we have genes. So therefore, as a result of that, mitochondria have its own genes. So since it have its own genes, it means that it can produce its own protein using its own genes. Remember from the concept of central dogma of molecular biology. So there are genes there that can produce a protein specific for the functions of the mitochondria. So now the question is, how do we inherit mitochondria? So usually the mitochondria or the maternal mitochondria or the mitochondria DNA is only inherited from the mother. It's only inherited from the mother. It's never, or it can never actually, or it's never inherited from the father. So mitochondrial DNA is a maternal inheritance. So we inherit mitochondrial DNA from our mother. So generally, if a mother is having a defective gene in her own mitochondria, then that gene will be passed on to the offspring. The genes will be passed on to the offspring. So all the offspring, whether male or female, will all have the mitochondrial DNA from the mother. So our mitochondrial DNA is actually coming from maternal inheritance. So you cannot get mitochondrial DNA from your father. So that is why sometimes in determination of maternity is very easy and simple. If you want to know who is your father or who is your mother, it's simple. You can just go ahead, get the DNA from the mother and then get the DNA from the child, and then you now run to a gene electrophoresis. You can easily identify the pattern. So that is mitochondrial inheritance. So as we continue with the lecture, we are going to look at how the other causes of metabolic or inborn error of metabolisms. So like, for example, what are the diseases that are associated with the mitochondrial inheritance? We have what you call Leber's hereditary optic neuropathy. So in the electron transport chain, we have what you call a complexes. In the electron transport chain, we have complexes. We have complex 1, 2, 3, and 4. So in the electron transport chain, if there is a defective complex 1, if complex 1 is defective in electron transport chain, then it leads to blindness and cardiac conduction defect, and it's usually occur between the age of 12 to 18 years. So if you have a child of between 10 to 18 years, if there is this mitochondrial defect, especially a defect in complex 1, then there is a likelihood that the of course, this complex 1 are enzymes. They are enzymes. So if it is defective, it means that there is a likelihood that it usually affects a child of between 12 to 18 years. So even if a child is having that defective genes, the disease doesn't show until when the child reaches a stage or reach an age of 12 years. Then we also have Lesch syndrome. Lesch syndrome is also a metabolic disorder that is associated with the deficiency or with the defects in complex 1 of electron transport chain. And of course, this disease is associated with the movement disorders. So it usually affects the movement of a child. Then there is also what you call mitochondrial myopathies, which is also a defect in mitochondrial genome, which will lead to the mitochondrial myopathies such as myoclonic epilepsy and myopathy dementia. So this is actually the problem that is associated with this, and the problem is also associated with a defect of enzymes 1 complex. So in enzyme 1 complex is also associated, or the defect in complex 1 is also associated with lactic acidosis, strokes, and seizure. So this is actually the problem associated with mitochondrial inheritance. So if your mother is having a problem with the mitochondria, maybe any mutation in the mitochondria, then the mother can pass on that gene to her child, and it's not usually coming from the father. So then the next thing apart from the mitochondrial inheritance is recessive or autosomal recessive inheritance. So the next thing that we are going to look at is the autosomal recessive inheritance. So in the autosomal recessive inheritance is a pattern of inheritance, and if a parent has an autosomal recessive trait, so usually it doesn't show as symptoms. Like for example, a situation where there is a sickle cell patient or a sickle cell carrier, we know that in the carrier the sickle cell is actually AS. It means that the patient or the person have a gene or the scalar, but it shows no sign and symptoms. There is no sign and symptoms. So in order to pass it onto their children, so both parents need to carry the traits. So before actually the parent can be able to pass that kind of inheritance, so both the parent must be a carrier. Like for example, in this case, look at what we have. You see that here we have capital N, small n, capital N, small n. So it means that the capital N is dominant and the capital N is dominant, while small n is recessive and the small n is the recessive character. So that is why, because of the capital N is dominant, so in any case where, like for example here, when this n cross with another n, like for example this n cross with this n, that is why we have capital N, and capital L is normal. And then we also have capital N cross with the small n here, and that is why we have capital N, small n. And then small n here cross with capital N, that is why we have capital N, small n. And then small n also cross, then we have small n, small n. So because the disease is autosomal recessive inheritance, so it means that the inheritance can only appear when the recessive character crossed with another recessive character. That is when you will have the abnormal allele. So for example, now we have small n, small n. So this here is a mistake. So it's supposed to be small n. So small n and small n is the recessive abnormal allele. So as a result of that, you see when we have autosomal recessive inheritance, you see when we have capital N, you have normal. Then when you have capital N, small n, you have carrier. Then capital N, small n is also carrier. Then small n, you have the affected individual. So in the case of these recessive characters, usually an offspring have an allele of small n. That is the only situation where you will have the affected individual. So, and then the next thing that we are going to look at is the autosomal dominant allele, how the autosomal dominant allele also associated with the enzymes defect in an inborn error of metabolism. So you see in the autosomal dominant inheritance, autosomal dominant inheritance is one way that genetic trait can be passed from parents to their offspring. And when a trait is autosomal dominant, then only one parent need to have an altered genes to pass it on. So if one of the parent is having that, altered gene, then it can easily pass it. Like for example, look at it here. The capital D here is the dominant. So you see this is the parent. So from the father or from the parent here, this is the father DD. So you see the capital D, small d. So because the capital D is a dominant, so of course, in the case of the dominant inheritance or autosomal dominant inheritance, so if the father is having the defective allele, then there is a likelihood that any character that cross with this D, any character that cross with this D, it means that it will be affected. And that is why here we have capital D, small d. So we have capital D, small d, and then we have capital D also here cross with the small d, and we have capital D, small d. And that is why 50% of the offspring are affected with the disease. But when you have small d, because they are autosomal, it means that it is the normal allele. So then the next one is the sex-linked recessive inheritance. Sex-linked recessive inheritance. So now we are going to look at the sex-linked recessive inheritance. So in a sex-linked disease, usually are passed down through a families through one of either X or Y chromosomes. You know that we have this sex-linked recessive inheritance or sex-linked inheritance is usually occur or is usually passed down to the family through either X or Y chromosomes. So we have, it's also divided into two. We have sex-linked dominant inheritance, and we also have recessive, sex-linked recessive inheritance. So in a dominant recessive or in a dominant inheritance, it occurs when a variant gene from one parent can cause the disease. So if it is a dominant, so if one parent is having the disease, then the disease can easily pass on. So, but in the case of the recessive inheritance, both matching genes must be variant to cause the disease. So if only one gene is variant, then the disease does not occur. It is a mild. So someone who has the variant gene but no symptoms is called a carrier. So carriers can form or can pass variant genes to their children, but usually in that case, the disease do not occur because it is a recessive. So the sex-linked recessive most often refers to X-linked recessive because the disease is usually passed on through X chromosome. So X-linked recessive, so the sex-linked recessive can also be considered as a sex-linked recessive inheritance. X-linked, it can also call X-linked recessive inheritance. X-linked recessive inheritance. So usually this X-linked recessive inheritance disease is mostly occur in males. So males have only one X chromosome, so a single recessive gene on the X chromosome will cause the disease. So the Y chromosomes is the other half of the XY gene pair in the male. So however, the Y chromosome doesn't contain most of the genes of the X chromosome. So because of that, it doesn't protect the male. So diseases such as hemophilia and of course muscular dystrophy occur from the recessive on the X chromosome. So what we are saying here is in the X-linked recessive inheritance or sex-linked inheritance, like for example, now look at it. This is a normal female gamete, and this is the affected male gamete. So you see, you have X and X. So if X and X, you only have a carrier daughter because there is this X. But in the case of, okay, now X cross with Y. So we have XY. So this is a normal son. So we have also, if this X cross with the Y here, we also have XY. So it is a normal. But in a situation where you have XY, where you have the defective X with, you have a defective X and Y. So you see, because this is the defective or this is a carrier X, and of course man have only Y only have XY, it doesn't have XY. So that is why the disease is usually occur in men. It's usually occur in men. So if this XY, affected XY, is crossed with Y, like for example, this is of course the affected male gametes. It is affected male gametes. So usually sex-linked recessive inheritance, it only affects men. And of course, female usually become a carrier. They usually become a carrier because of this X chromosome. So these are actually two ways in which the inborn errors of metabolism occur: through sex-linked recessive inheritance, through also autosomal dominant inheritance, autosomal recessive inheritance, and then mitochondrial inheritance. So then the next thing that we are going to talk about is the inborn errors of carbohydrate metabolisms, which we are actually going to look at it in the next video. So ladies and gentlemen, if this is the first time you are coming to my YouTube channel, please subscribe. Thank you.